This is episode 300 of the Stem Cell Podcast, Autologous Cell Therapy, with one of our favorites, Dr. Jeanne Loring. Hello, everyone. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen here, 300 episodes in, you should rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, for the tricentennial, we have Dr. Jeanne Loring, who's from Aspen Neuroscience, and she's on the podcast to talk about her work on autologous iPS cell-derived therapies for Parkinson's disease. We've also got our usual roundup of recent highlights and stem cell news that's coming right up.
But first, we'd like to remind our listeners about Cell Therapy News, a free bi-weekly newsletter brought to you by Stem Cell Science News, summarizing the latest research, news, jobs, and events in stem cell research. Use Cell Therapy News to stay current with the latest cell therapy, gene therapy, and regenerative medicine research. Subscribe at www.celltherapynews.com.
All right, guys, I got some a little bit different here to kick off the roundup today. I like the format of these matters arising, or brief communications arising that have been in the Nature journals over the last, who knows how long. It's been years now, because I like the idea that allows you to revisit old stories in a format that isn't just hating, or like Pub Peer maybe, which is a lot of finger pointing without really a chance for the authors to defend themselves or to defend their findings or conclusions in a unified forum, right? That is like refereed. So I like the format of the arising, but typically you get like the dissent and then the response from the original author, right? In this case, we have a really unique case where we have a matters arising, which was more like a post-publication review and ultimately retraction, but in this case, not because of like scandal or fraud or anything, but really just about maybe perhaps, I think the claim being made here, although the original authors don't agree, and we'll get there. But I think this is a unique format because we had this post-publication review and then a kind of questioning of whether the validity of the conclusions and then a retraction on the part of the journal, which I guess ideally that's the way the system works, right? Because all these things we look at, it seems like they're set in stone because they came out in print. But here we know years later that many of the stories that, even though they've been retracted, some of them, there's a lot of stories out there that are, not exactly true. Or maybe the conclusions were a little bit, I don't want to say far-fetched, but maybe aggressive, right? So here's the story. This is in Nature just recently. And the original story was from Richard Lulab, who's at Cincinnati Children's Hospital. And they generated this single-cell RNA sequencing atlas. This is back in 2022. It was a fetal human cerebellum. And the takeaway was that they identified this previously uncharacterized cell type that they designated a transitional cerebellar progenitor, right? These TCPs that had a transcriptional similarity to medulloblastoma, right? So the exciting idea there was that now you had a kind of a link and developmental origin potentially of this tumor, which is, a terrible malignant childhood cerebellar tumor. So the data, and we'll come back to their assessment of the data, but the Paul Northcott's group, who's at St. Jude Children's Research Hospital, they did a reanalysis of those same data and then went a little bit deeper. So I'll elaborate. They got the gene count matrix from that original report. And just to give a little more detail, this was fetal human cerebella of 8 individual donors that were representing 8 developmental time points, right? And from that data set, the original authors from the Lulab, they described these TCPs as NSC derived, so neural stem cell derived cells that give rise to granule cell progenitors, unipolar breast cells, and Purkinje neurons. And the peak of these TCPs they identified at this 12-week post-conception time point. That's when these TCPs peaked. But here's the thing. In the reanalysis from Northcott's group, they found that of note, I think most notably, 90% of the cells that belong to this TCP population in the whole data set were obtained and originated from a single donor. Okay, this was a donor at the post-conception week 12. And within this donor, and also across other samples where the other 10% were found, these TCPs showed both the fewest unique molecular identifier, these UMIs, as well as genes detected amongst all the samples, right? So that was like the first red flag in the cursory analysis by Northcott's group. So they went a bit deeper and did the whole reanalysis from the FASTQ files using the same parameters as the publication. And in their analysis of specifically this post-conception week 12 sample that accounted for most of the TCP cells, they showed that there was a high background fraction, reduced droplet efficiency there, and other parameters that ultimately were consistent with a high abundance of ambient RNA. So ambient RNA is free-flowing transcripts from just light cells that contaminate endogenous gene expression profiles. Right? They're not specific to the cells you're talking about. They're just in the droplet. So even in the other cells, as I alluded to earlier, these TCPs, the remaining 10%, they also showed low nuclear fraction, low UMI counts, reduced detected genes, high background fraction, and overlapping with empty droplets, which suggests that even empty droplets had the same transcripts, again, contributing to this idea of both low quality cells within the droplets where they were bona fide detected, but that ambient RNA was non-specific to that cell type, and in fact was general to the entire population, non-cellular ambient RNA again. And then when they take out that population, so the post-conception week 12 time point accounted for 90%. When they take that out, the remainder of that TCP structure from the other samples, it falls apart. It no longer is distinguished as an individual cloud, and it becomes integrated into those unipolar breast cells and those GCP cells, which I forgot, the granule cell progenitors. So these unique TCPs essentially melt into already characterized populations, and then going even further, because you can't just throw stones. You really gotta provide a kind of positive negative. They went into other independent fetal cerebellar atlases, in this case encompassing post-conception week 7 to 20, and showed with a rigorous and exhaustive characterization of those cells that they were unable to corroborate the presence of this TCP-like population. And the markers that were uniquely expressed in that TC population identified the Lu lab showed no specific expression within that data set. So ultimately, the journal, immediately after this report, retracted, or upon publication of this report, also retracted the previous study, and the original authors from the Lu lab did not agree with this retraction, and they did provide a response to firm up that data. But notably, that response is published on bioRxiv, not as a kind of response within the same journal, as is typically is the case for these matters arising. So you wonder if it's the consensus there that's reached in post-publication review that, yeah, sure, you have a response, you're entitled to your response, but your response it does not do much to convince us of the original claim, and so it's just a retraction. So I hadn't seen that before, Arun. I thought it was, I don't want to call it scandalous, but it's hard, I think, for any result to be challenged. It can create some drama, so to speak. But I thought this was really constructive, and I also want to note that this is done with great care. This matters arising, the original dissent was received more than 2 years ago, so in March of 2023, so within a year after the original publication. But it took 2 years for the editors to be convinced that this final outcome of a retraction and not publishing the response in the same journal issue was merited. So a unique case, and I would say, all in all, a positive narrative of a negating a result.
Yeah, I like the way you kind of spun it there at the end. I'd like to hope that this was, more of the common situation across science, but you know as well as I do that this is a bit of a rarity. And I think part of the reason that these folks decided to challenge some of the initial findings was because the initial findings were quite a bombshell, right? They were mentioning this transitional cerebellar progenitor, this never before identified progenitor cell type in the brain, and that's big news. The initial study was, of course, very high profile as well. It was initially a Nature paper, but it also makes you wonder how many of these situations don't happen, right? Simply because the study is not, quote, high profile enough to warrant a rebuttal, I suppose. This is a really, certainly extremely hot topic. I wish, this happened more often. That's what I'm trying to get at here. I'm so thankful, honestly, that the folks here on this rebuttal, the Norcott lab, Kyle Smith, really, in addition to just addressing the initial paper, they turned this, like you said, into a completely novel independent study, validating or disproving those initial findings. So it took a lot of work to do this. Like you said, multiple years. This actually reminds me of another situation that was many years ago when I was in grad school, where folks were demonstrating the efficiency of the direct reprogramming process that we've talked about here on the show, and in particular going from fibroblasts to cardiomyocyte. And I think some of the work was initially coming from Deepak Srivastava's lab in 2010 or something like that. And I remember my grad school mentor, Sean Wu at Stanford, he actually published a pretty high-profile circulation research paper, basically as a rebuttal to that direct reprogramming process in cardiomyocytes, saying in the title, inefficient reprogramming of fibroblasts directly to cardiomyocytes. So, but he did the exact same thing. That paper was a multi-year study that was showing that the initial observations were not what they seemed. And in that case, it was just validating the inefficiency of the process. This study is showing that the initial observations may have been an artifact, right? So this is something that honestly we need more of in science, but I'm a little cynical in that I know scientists, of course, academic scientists in particular, we have our egos and we hate to see our findings challenged in this sort of way. But given the issues with reproducibility in science, we need to see this more often, don't you think?
Oh, absolutely. And, I think it's something that we are going to see a lot more of, because we're in this era now of all this work was done in silico, right? This was existing data that was reanalyzed and cross-referenced with other data sets. So no pipettes involved. And I think that there's a latency here, because I don't want to say that single cell sequencing has grown up, right? But it's been more than a decade, right, at this point. But still, I think that the ubiquity of single cell sequencing now, I would say at the point of publication for this, you could publish a single cell data set based on the fact that it was a data set and draw some conclusions. Now, not so much anymore. So I feel like there's a latency where a lot of those studies ought to be, as you said, reused. I don't think a lot of them will be, as you alluded to there, because sometimes I've been close to, like you, I've been close to some of these things. One in particular. And what I recall about that is that there's so much animus, as you're saying, like it's snarky. Like the reason why the initial dissent came is because that guy hated you already. And so he was looking out and looking for a reason to take you down, you know. Whereas I feel like we should be living in a scientific era, which probably will never come, where we welcome further scrutiny of our results, because for sure the capability is there, right? We're able to revisit all these data sets. And I'm wondering if there's a way now using these in silico measures to try and glean some of the truths or artifacts of experimental results, which maybe is a bit, we're not quite there yet. But you wonder with the modeling and the inference that you can draw from these, how accessible now the revisiting of old results is for all of us. So I don't want to say exciting times, but things are changing for sure.
Things are changing, and tools like, of course, AI can help enable some of this fine-tooth combing of some of these in silico data sets, and it'll make folks' lives easier in some ways. But a broader discussion, which is probably worthy of a topic of an entire episode, is that, you know, scientists, we're not as objective as we'd like to be, right? We're still people. Science is not entirely disentangled from, say, politics or from personalities, because people are doing this science, right? It's not AI who's actually publishing these papers, as far as I know. Yet. And people have egos. People hate to be proven wrong. And, you know, we have our strong feelings, and we have our haters, and we have our lovers too. We have friends in the fields. We have scientific enemies, of course. It's all really part of the game. But big picture, yeah, once again, shout out to these folks, the Norcott lab, Kyle Smith, for leading this really important work, published in Nature. So moving on to actually another study coming from Cincinnati Children's, or affiliated with Cincinnati Children's. This is a friend of the show who we actually caught up with at ISSCR in Hong Kong. That's Ming-Sha Gu, who was at Custom, this amazing organoid research hub over there at Cincinnati Children's. She started her lab there, I think was there for a few years. Has published a number of high-profile papers in the area of organoid development, especially when it comes to cardiac and lung organoids and various different applications and vascularization and so on. Turns out Ming-Sha is my neighbor now. She actually has just moved down here to UCLA, and I'm actually trying to get her to give a talk here at Cedars-Sinai. We're just 15 minutes down the road, 30 minutes depending on traffic. So this is a cell study coming from Ming-Sha's lab. Also on this paper is Yu-Shrike Zhang, who's an amazing bioengineer at Harvard, and I know Ming-Sha has collaborated with them quite a bit. He's actually also done some work related to space research, and he's run into my circles as well, which is kind of cool. First author here is Yifei Mao and Nicole Peck, I believe, who was a grad student, trainee in Ming-Sha's lab, also led this effort. Co-development of mesoderm and endoderm enables organotypic vascularization in lung and gut organoids. And I think this is, you know, exciting for a couple of different reasons. One, we were talking about vascularization, and vascularization is the new hot topic in organoids. And of course, you want to make these organoids more complex, being able to flow nutrients organically and develop that vascular network within the organoids. That's all the rage these days. But another reason why this is a hot topic paper, independent of vascularization, is the co-differentiation, right? You're basically differentiating multiple germ layers simultaneously into a single system, into a single organoid system. And I know other folks at Cincinnati Children's are working on this as well. We actually have another Cincinnati Children's paper in the Roundup. Here, they're co-differentiating lung and gut organoids. So vasculature, mesenchyme, these are things that exhibit, of course, distinct organ-specific characteristics. And they are adaptable to the local physiologic needs. And we've talked about this on the show. Vasculature, it's not one size fits all. It's not just plumbing, boss, as your mentor has said many, many times. Vasculature is very organ specific. You know, you got fenestrated, you got all different types of vasculature, brain microvascular, cardiac vasculature. It's different. It's not one size fits all. So the development of vasculature and mesenchyme and all this good stuff is shaped by the microenvironmental and cell-cell interactions from early development, as you would expect. And if you want to recapitulate that process of vascular development or organotypic, organ-specific vascular formation, you have to kind of recapitulate the process of development, right? So you got to co-differentiate, which is what they did here, mesoderm and endoderm within actually the same spheroid, same three-dimensional pluripotent stem cell derived tissue to what they did, generate vascularized lung and intestinal organoids from iPSCs. Turns out that BMP, bone morphogenic protein, of course, one of the really critical signaling axes in development, was really important for fine-tuning the right endoderm to mesoderm ratio, which is I think probably the most critical step in actually generating the right amounts of endothelial epithelial progenitors within those intestinal and lung organoids in that grander super organoid, I suppose. And then, they did the single cell. Of course, you got to do the single cell with the organoids. And they were able to confirm that there were organ-specific gene signatures of endothelial cells and mesenchyme. And they also identified key ligands that were actually driving the endothelial subtype specification within the lung, within the intestinal organoids. The endothelium did what it was supposed to do. It exhibited tissue-specific barrier function. I mean, something that I've delved into in my lab is looking at endothelial barrier ability, and it's different whether you have cardiac endothelium and for that sort of vasculature, or brain microvascular endothelial cells, which have a higher transendothelial resistance, so they have a stronger barrier. So different endothelial subtypes have different barrier capabilities, and that's what they're able to further confirm using this particular analysis. Also looked at cell diversity in the organoids, alveolar formation on an engineered lung scaffold. So there was a bioengineered element to this. Like I mentioned, Shrike Sang is a part of this paper, amazing bioengineer. And then part of the reason that this ultimately went into Cell was they did the transplantation work. So they transplanted these combo organoids into mice, and the organoid vasculature was able to integrate within the host circulation, which is actually able to preserve the organ specificity and promote further the organoid maturation. And another thing that we've talked about a lot here on the show is integrating these engineered vascular networks into the host architecture to actually further drive their development. That's exactly what they did. And then they did a patient-specific mutation, patient-specific disease modeling element of this at the very end, where they uncovered abnormal endothelial epithelial crosstalk in patients with a FOXF1 mutation. So really neat system developed by Ming-Shi's team here, multi-lineage organoids. It's multi-lineage is really the way of the future for in vitro modeling. You got to look beyond a single germ layer. You got to look beyond just the mesoderm, for example. You got to, especially if you're trying to study development in some capacity, you got to sort of better approximate how these things are actually happening in vivo. And they're able to do that by, again, fine-tuning BMP to some extent with this co-endothelial mesoderm differentiation to create this really unique model of development here.
Yeah, I mean, I don't want to go crazy here, but I feel like this is a pivotal, seminal, watershed kind of study, and the study has a lot. You synthesize it very well, but I think it's representative of a sea change in how we think about these things. So yeah, all props to Ming-Jia, but not this alone, but this and other studies, I think, signals a shift of thinking away from, the assembloid idea. I think as it was born by Sergiu Pasca, I think it was amenable to this idea of connecting things, right? Because you do have these nodes. It makes sense in the context of neural, but in the context of vascular, and we've talked about this so many times, right? The idea of making an assembloid, a vascular assembly, where you take a vascular organoid and then whatever organoid and put them together, doesn't really capture the native physiological vasculogenic process, right? It's something that happens, as you said, it's integrated into each developmental event. Each germ layer, each tissue enlage has its own rudimentary vasculature there that grows along with it, and it actually is instructive, right? A lot of key findings. I think now we're moving toward this idea of like that. We got to find a word for it. Integroids? I don't know. But the point being is that you have an assembloid where it's not putting things together, it's growing things together. And the irony there, I think, is that it gets you right back to kind of square one in terms of, isn't that an embryoid body? But I think now we have a level of resolution and controlling the signaling and morphogen gradients, perhaps, hopefully, with engineering approaches and, all that we've learned over the past 2 decades plus. That allow us to make, something that's less embryoid body, more embryoid model, embryo model, without the whole embryo, just, tissue enlarged model. And I think that we're there. And I think this is the among this first wave of studies, particularly in the case of the vasculature, because I think that is also such a bottleneck. Because if you can unlock the vasculature within a tissue-specific organoid, that provides a lot of angiography cue. I don't want to say perfusion, but just think, maybe then it provides a medium, a substrate for immune cells, which can be very instructive for developmental events. As we look in development, what always tracks right alongside the vasculature? The neural pathways, right? So, which is also really important for generating organoids and incorporating the neural component, but also can be instructive just in terms of tissue morphogenesis. So I think that maybe this is the integroid that, as Sergio Pasca has gone with the 3 and then the 4 components putting together, maybe we're adding more and more cell types, right? So maybe the immune next, maybe the neural, and then we're going to get towards something that's really just a little piece of tissue in a dish. And that's the dream.
Yeah. And that exact point makes me reconsider my entire approach to in vitro modeling, right? It's like the whole objective of our models has been trying to faithfully recapitulate development in vitro using these, like in my example, just artificial cardiac organoid systems. But the actual embryo models are becoming so advanced that should we just all use those instead, right? Oh man, now I'm having an existential crisis here. Thanks for that, Daylon.
Oh, my bad, man. Well, you make a good point, but I think at the end of the day, you can lever the experience you have with the nascent cardiac signaling apparatus and just incorporate, I think, more of the elements. And that's the thing. I don't think we're going to start from zero. I don't think we're going to start from the 3 major cell types in a pre-implantation embryo and go from there. I think that we'll reach the sophistication where we can, move the chip ahead a few weeks in developmental timing and start with these organ enlages that then roll downhill into something super complex and much closer to physiology. So I'm sorry to throw you for a loop there, Arun, but you're going to be just fine.
All right. Good to know, man. All right. Good to know.
Moving on to a little quickie I have here, which is a resource about the blood. I mean, you talk about vascular. It's only fair for me to get into the blood here. It's momentum. And this is a resource in Nature Cell Biology, which had a massive undertaking from many investigators, jointly supervised by George Boucher and Nina Cabezas Walscheid. Sorry for any mispronunciations there. They're at the Max Planck. And the idea here was to leverage tech to generate a data set with high resolution. And then, of course, given that this is a high impact paper in Nature Cell Bio, to actually demonstrate proof of concept of how this data set can be useful experimentally and perhaps even clinically, right? And what we're talking about here is hematopoietic stem cells, which are very rare. And their rarity and scarcity within the body is linked to the fact that they are quiescent, right? And under homeostatic conditions in the body, they're quiet little sleeping beauties. And that's so that they can be resistant to cytotoxic stresses and DNA damage and any of the events that can undermine or adulterate their quality with proliferation. Allows them to maintain this healthy stem cell pool that can be mobilized in response to whatever insult. And this quiescent state, it's primarily associated with glycolysis, so this resting metabolism. But HSCs in their quiescent state also exhibit some flexibility and utilize alternative metabolic pathways, including fatty acid oxidation, which is going to impact the level of lipid consumption within the cell. Now, metabolomic and lipidomic studies and data sets generated in the past few years have increased in the resolution and sophistication, but they're still limited in the context of hematopoietic stem progenitor cells, particularly lipidomic studies, because the low input, right? There's few of these HSCs and also not a ton of human bone marrow samples to get the primary HSCs from. So that's a major challenge. This story is really one of optimizing the methodology and the workflow to generate a data set with really low input, but a data set that has high resolution. And the impact of this and the rationale for generating this data set is clear. We talk about it all the time. The holy grail of hematopoietic biology is how do you generate a metabolic milieu that enables long-term ex vivo expansion without exhausting the hematopoietic stem cell pool, right? Because that would be a boon for so many therapies, not to mention, HIV cure, for example, as an out-of-the-box application of unlocking the unlimited potential of hematopoietic stem cells. So a data set, the rationale for a data set is high. And there's a lot of tech in this resource, but the bottom line is that it's there for you to enjoy and to browse and to apply the same workflow towards whatever other scarce data set. And the way they prove the principle here is by generating the data set, and then I'll come to the conclusions and the application there. So first, they got 85 human bone marrow samples across the study, so a very broad range that encompassed young, old, as well as AML patients, right? So that was kind of the platform, is to look at the good, the bad, and the ugly, so to speak. And so they found a lot of trends that were in terms of the metabolism and lipid metabolism, particularly that went across that range. And the specific one in terms of proving principle, they found that choline levels were really high in the young hematopoietic stem progenitor cells. Those declined with aging, and in leukemia they were particularly low, even lower than in aging. So this seemed to be a progression that was linked to the fitness of those hematopoietic stem cells. And then, of course, they show that if you supplement the cells with choline, the metabolite choline, that you enhance their stemness. So a little, experimental result and a lot of data set here, but the totality of it for me is twofold, manyfold perhaps, but at least in terms of obvious application, yeah, sure, we have a data set here that I think a lot of people would be happy to dive into to ask their specific question, to either test or generate a hypothesis. And also the validation here. We had another story a few episodes back that also talked about, I forget what the metabolite, well, I think it was glutamate in that case. We were joking about Chinese food and MSG, but another case where like a very straightforward nutritional supplement can have potentially an outside effect on your stem cell pool, in this case in the hematopoietic, maybe in other stem cell pools as well throughout other tissues in the body. So I was glad to see another, I don't want to say the validation of the original result, but another story kind of linking the metabolism and modifiable metabolism at that to stem cell pools and, by transitive property of quality there, to health and clinical intervention. So a little resource that I think is going to have a big impact.
Yeah, I like that. I mean, this is omics to the max, but also it's a tech story, right? It's low input targeted metabolomics, which is enabling these observations to be found. And I love the endpoint there, right? Because that's that identification of choline supplementation that could fuel lipid production in HSCs. I mean, that's immediately testable, either in various in vivo systems or even I could take some choline myself. I don't know. We'll see we can see what happens. I'm not endorsing that, but hey, people are gonna attempt these things, I suppose. Yeah, these are some of my favorite types of studies, which do the deep omics level analysis, and we've covered a lot of these Atlas papers, which do the massive omics, single cell omics, so metabolomics in this situation, but it's the it's the observation at the end, it's the hypothesis generation at the end, which really excites me, because I'm just an old school scientist, I suppose, right?
You are old school, Arun, but you are a young king. And I mean, I will say that I love your point about the testable element, right? Because there's so many stories where like, oh well, I gotta not if in order to test, I gotta knock out a gene, or it's druggable, but like if I drug that, I'm also going to kill the patient. Like there's all these things that seem to have their limitations. So like, yeah, the finding is valid, even though it may not be practical clinically. Here, it's like you're exposed. If you're this story, you better be confident in this result because people are out there doing some choline tests. And just FYI, my friend, liver and kidneys, or organ meat, very high in choline. And if that's not your bag, like me, you can also get into the shiitake mushrooms, broccoli, cauliflower. So there's a chance for us. We're going to have to eat some General Tso's kidneys or something to get the combo of like choline and glutamate, live forever.
Oh man, I love that. I do love shiitake mushrooms. So that's an easy in for me right there. Easy fix. Moving on to the last paper of our roundup here. I alluded to it a couple papers ago. This is also coming from a custom person. That was a terrible description. It is coming from a researcher at Custom at Cincinnati Children's, an amazing organoid research and development hub. And this is Takanori Takebe, who needs no introduction at all. He's of course been such a world leader in the field of modeling various pancreatic disorders, various liver development phenotypes using all different types of organoids. His career has taken off like a rocket over the last 10 years and has been very prominently featured at ISSCR. Of course, he has a Japanese origin story, but he's shifted over to Cincinnati Children's many years ago. But I think has some pretty strong connections with folks in Japan still, and I'm pretty sure he's going to be at ISSCR in Kyoto next year. I can't wait for that meeting, man. I'm for sure going. But I'm sidetracking here for a little bit. This is going back to this really cool Nature Biomedical Engineering study. We don't cover Nature Biomedical Engineering too much here on the show, but they got some really cool papers here utilizing scaffolds and all these engineered approaches to take your organoids to the next level. And Mingsha did that a little bit in her study a couple papers ago as well. So here they are differentiating pluripotent stem cells into CD32B putative liver sinusoidal progenitors. So the title of the paper is Self-Organization of Sinusoidal Vessels in Pluripotent Stem Cell Derived Human Liver Bud Organoids. And like I said, Dr. Takebe has been working in this area for a long time. Sinusoidal vessels, as you probably know, are really critical in the liver. I mean, they have these unique fenestrated structures that are really important for the filtration of the metabolites and all that good stuff in the liver, helping liver do its job, right? But the induction of, like what we talked about in Mingsha's paper, the induction of tissue-specific vessels in these in vitro living tissue systems remains challenging. You have to recapitulate a lot of the in vivo developmental pathways, sure, right? And Mingsha was able to do that by modulating BMP signaling, for example. They took more of an engineering approach. They devised an inverted multi-layer air-liquid interface culture with hepatic endoderm, septum mesenchyme, arterial, and sinusoidal quadruple progenitors that self-organize to generate and sustain hepatocyte-like cells. And they're neighbored by divergent endothelial subsets, which have like all the markers that you might expect of these sinusoidal endothelial cells. I'm not going to name all of them. There's like 15 markers that they actually use to validate the cells that they're making. And life comes down to Wnt signaling, as we know. So they found that Wnt2 mediates this sinusoidal to hepatic intercellular crosstalk, which actually potentiates the differentiation of hepatocytes and the formation of that quintessential branched endothelial network that's found in the liver. And then they did some deeper imaging to actually show that the iPS-derived liver sinusoidal endothelial progenitors actually develop fully perfusable vessels. That's powerful, right? It's more than just making these random smidgens of CD31, CD144 positive, quote, endothelial cells and calling it a day. Is showing the functionality. They're actually creating lumen structures, perfusion, like what endothelial and vasculature are supposed to do, right? And then they did a little bit more work here, and organoid derived hepatocyte and cytosolic derived coagulation factors was able to correct this this defect of clotting time with different factor deficient plasma. This is looking at primary blood samples. That's pretty wild. And then kind of wrap things up with a bit of a disease model, which was a model of hemophilia, of all things. So this correction approach was able to rescue this really bad bleeding phenotype in hemophilia A mice in transplantation after they actually transplanted the system into the mice. So another way to engineer your organoids to the next level. And it's engineering oriented, perhaps more so than Mingsha's paper, but this really gets to the point of vascularization. That's what I'm trying to hit you guys over the head with for this particular episode. It's more than just making your organoids now. You got to make them better. You got to make them functionalized. You got to integrate them perhaps with the host vasculature to see how you can further enhance their maturity, like what Mingsha did. And importantly, it's making your vasculature functional. Right, that's what vasculature is supposed to do. It's a conduit. It's more than just plumbing, sure, but it is still plumbing. So you got to be able to flow stuff through the plumbing, and that's the importance of developing stuff like lumens, for example. Right. So a really interesting study from the Takebe group. I think more bioengineering oriented than their lab is traditionally known for, but I mean, Dr. Takebe can do a little bit of everything, I suppose.
I mean, he does it all. This is an engineering story, 100%. I mean, there's a lot of cells here, but if you just looked at the videos of this paper, of this story, you blow your mind. Particularly the supplementary video 4, where you get the visual on this hemophilia correction element, where you actually see like the blood flowing and then clotting. I just love, there's nothing like seeing how they test these things. You know what I mean? How do you take a system, a cell system, and see if you can correct hemophilia with it? And the system that they use here is brilliant, and also visually very compelling. So I invite all you guys to take a look. But yeah, the story in its totality, just like the other one, this integrated approach, and now addressing the question that now I feel like we thought about very naively 20 years ago when we were first making endothelial cells in my case, and we said, oh, wait, when this will apply for hemophilia, we'll make liver-specific endothelial cells and blah, blah, blah. But did we ever get any factor expression for any of those ECs? No, and we gave up because we weren't even close. Now we see all the work it takes, the phenotyping. I mean, the exhaustive phenotypes of these. It shows both the work that goes into it, but also the specificity and singularity of these progenitor cell types that exist. Really blows your mind, makes you realize that although we have these specific milestones that we talk about in development, this progenitor, that progenitor, this pathway, that pathway, we're really only seeing a fraction of the picture. There's so much left to learn, and the subtle differences between all these cell types along their developmental hierarchy, as well as across the different types of progenitors, I think is so fascinating and will certainly keep me busy just in my little corner of reproductive biology for the rest of my career, for certain.
Yeah, it's the visuals that are so striking and so appealing. I mean, I'm a simple scientist. I love seeing things, and seeing is believing, right? And we have had so many striking examples of organoids that are doing what they're supposed to be doing, whether it's alleviating this hemophilia phenotype, or generating tears in vitro, or generating hair in vitro, or cerebral spinal fluid, or contracting. I mean, these are all amalgamations, approximations of what's actually happening in vivo. But if nothing else, it's just really visually very appealing to see these things happen live in front of your very eyes, right?
Feast for the eyes. And I'm very appreciative of your emphasis on the vasculature in this show, Arun. It really was a literally beautiful display that you did. Moving on to what will certainly be a beautiful discussion, fascinating and deep and varied with Dr. Loring in just a minute. But first, I got a quick message from STEMCELL Technologies. Neuroscientists looking for more predictive power in their disease models are increasingly adopting human pluripotent stem cells in their research. STEMCELL Technologies offers products, protocols, and training to support iPSC-derived neural models. Explore their collection of technical videos and webinars on neurological disease modeling by visiting www.stemcell.com/neural-disease-model. All right, you guys, on today's show, we have a real special friend of the podcast. Every time we run into her at the ISSCR, we get to talking, and then, the minutes pass joyfully. So we're going to share a little bit of conversation today with the illustrious Dr. Jeanne Loring, who is co-founder of Aspen Neuroscience, also Professor Emeritus at Scripps Research Institute. Jeanne Loring is a scientist, an ethicist, and a pioneer in stem cell technology and therapy. She founded the Center for Regenerative Medicine at Scripps Research and has founded biotechnology companies, not just Aspen, but she's been around the block, including, of course, Aspen Neuroscience, which is venturing perhaps the most ambitious efforts of all the companies that she's founded. She's also published more than 250 scientific articles and 2 books on pluripotent stem cells and genomics, epigenetics, rescuing endangered species, all pretty much centered on stem cells, and not just on Earth, but also in the International Space Station. So she's really all over the place, not just here on Earth, but interests beyond. Also a mentor and science and business advisor for stem cell researchers and companies, especially startup biotechs. So yeah, Jeanne, she wears many hats. And today she is putting on her communicator hat to share with us and all of you her many adventures and insights surrounding our work. Jeanne, thanks again so much for joining us today.
Oh, you're welcome. It's always a pleasure. Every 100th time, I hope you're invited.
Yeah, maybe we can turn that into a thing. Have you back on for the 400th episode as well. Yeah, we can pencil that in. Anyways, welcome back to the show, Jeanne. And it's so fun catching up with you, just because, you never know where the conversation might go. I mean, you have this eclectic collection of scientific interests, like Dalen alluded to, ranging from treating Parkinson's disease, sending research to space, which is, of course, something that I love to do as well, or trying to save endangered species. It's all united, I think, by your love for stem cell biology. So I guess let's start there. What drives you to pursue such a diverse range of scientific interests and actually be so successful in each of these pursuits?
Yeah, so I think the fault is that I fell in love with stem cells a long time ago. And that, just falling in love with stem cells, you start thinking, well, there's all these things they could do. And then I've had a really hard time focusing on a single thing. So the combination of stem cells and stem cells pluripotency and my interest in doing, new and interesting things, it just coincides. I mean, it's actually surprising that I'm doing so few things.
Well, I mean, it's a bit of a case of ADHD here, though I would call it attention deficit hyperproductivity disorder. I mean, you can't call it a disorder; that's a gift. And as we're saying, there's just so many places that we could start. We highlighted very briefly there in the jump. And there's no way we're going to get to all of it. So just selfishly, to ensure that I get it in, I want to begin with this, at least my questioning here, with the common interests that you and I share. It's a bit fringe, like some of your other interests, but it has outsized implications. I think you'll agree. I'm talking about efforts to share the northern white rhino, which is functionally extinct with only 2 non-productive females remaining. We talked about this a few times on the show with your boy Arun Seabass as well. I mean, we've highlighted this because we think it's important, but also and from the species preservation, just on its face, that's very important. But also the effort to generate gametes from white rhino iPSCs, I would argue, is seen by many as a testing ground for similar efforts to generate human gametes. And I've been pretty clear about my concerns regarding, on the show, at least regarding my concerns about applications of this approach in humans and the risk to germline genomic integrity. And some might even argue, I think, kind of maybe even in microcosm, that the risk of genomic drift may be a problem, you could argue, with any cell type that you expand ad infinitum in vitro. And we're talking here somatic, not germ cells. Like, at what point do those expansions and the diffs and all that cell culture undermine the integrity of the cells? I don't know. I think an open question. And you were recently a part of this effort to map the northern white rhino genome, ostensibly, I mean, directly, based around the idea that we need a reference, right, to ensure that the genomic integrity of any iPSC-derived gametes is preserved. But I want to ask you about that. Please share any details you like about that project. But I want to ask you more to the bigger picture there. If you have any thoughts about, like, what's the bar for safely allowing use of this approach in humans? And do you think, to that point that I kind of alluded to earlier, do you think there's any latent risk of the kind of genomic integrity issue with somatic cell treatments? And if so, like, what do we have to do in order to ensure the safety there? A lot of questions, but I mean, you got it.
No problem at all. So with very few exceptions, I think epigenetic and genetic changes happen when cells divide. There was it was a very sort of gross and broad-ranging idea, the bad luck cancer idea, that the cancers tend to form in tissues that have lots of cell divisions. I mean, it makes perfectly logical sense. The cells will evolve. We know they evolve. We've known this for a very long time. You have them in a culture dish. They're dividing. They keep dividing. iPS cells are especially guilty of this. As long as they keep dividing, they will accumulate mutations. The question is whether those mutations are harmful or not. And so the questions you have to ask are, because we're mosaics, we have lots of different sequences in our, I have not seen a comprehensive analysis of that, but I do know that, for example, among our white blood cells, they have different DNA synthesis, different DNA sequences, apart from all the rearrangements that they make. They just accumulate stuff as they divide. So doesn't seem like it's anything we can fix. It really is sort of a balance of how much DNA repair, how directed DNA repair is, how long the system has to repair the DNA before the next DNA synthesis comes along, and just keeps on doing the same thing, acquiring changes that stay. And then there's this natural selection, which happens in culture dishes, that if a cell does better than its neighbors, it'll take over the culture eventually. It's just a matter of time. So I think this is something that, I mean, I've definitely been beating the drum about this for a really long time, and not mostly unsuccessfully, because it's a raining on a parade kind of thing.
Yeah.
You say, yeah, great, good work, guys, except for, did you sequence yourselves? I've been doing that for so long. I mean, like, people that both you and I know, Arun, when I would ask them a question at a conference, when they gave a talk, and I said, would you sequence yourselves? And they would essentially say, well, why would I do that? So the answer is that eventually everybody's going to realize that the reason you have to do it is because that happens, and you can't stop it from happening. You can't pretend it isn't happening and not have it happen. So there's lots of implications. And I think for germ cells, the really hard thing, and I do advise a company which is trying to make germ cells out of human iPS cells. One of the companies I advise. I think the science is really good. The question is, how do you decide, and who decides whether an iPS cell derived oocyte, for example, is sufficiently without flaws? And we're not just talking about inherited things now. We're not talking about IVF. We're talking about something that arose while that cell was an iPS cell.
Mm-hmm.
Then how do we draw the line on whether that's a safe thing to use? And then the problem is, you will either find out very quickly because it's so devastating that the embryo won't implant or the embryo dies young, or the worst case scenario is probably that the embryo survives and you get a child born, and that child has some kind of brand new genetic defect. And that is not, it would, just imagine the scenario in which the child has a brand new genetic defect, but it doesn't manifest until their older age, and they've already passed it on to the next generation. We have changed all of humanity by doing that. So yeah, big picture, big thing to think about. Luckily, we're not there yet, so the decision doesn't have to be made yet.
Yeah, I mean, that's a scary proposition, that's for sure. But it's a conversation that's especially relevant as we're, developing cell therapies. It's such a golden age for iPS-derived cell therapies, for example. And of course, this is my segue into talking about the data that's emerging from Aspen Neuroscience, of course, the company that you co-founded, which is using iPS-derived dopaminergic precursors for treating Parkinson's. I mean, this is an incredible unmet medical need, and sorely needed, right? A cell therapy in this area, an effective cell therapy in this area. And we were at ISSCR not too long ago, where Andres Bratt-Leal from Aspen, I was actually presenting some data from the first few patients of the Aspiro study. This is that phase 1/2 trial of the company's iPS-derived autologous cell therapy, and it's an open label trial which was designed to evaluate the safety, tolerability, efficacy of these, intracranial implantation of these autologous iPSCs, derived dopaminergic precursor cells. Importantly, in the patients with moderate to advanced Parkinson's disease. So, as the co-founder of Aspen, I'm sure it was really exciting to see the trial progress to this point. So really just tell us about it, how you guys got to this point, and really what's next.
Yeah, I'll try to keep it a short story this time. We first got the idea in 2012. Actually thought about it for a long time. It was pretty, if you're a neurobiologist and somebody says you can make a cell that can replace a neuron that dies in a disease, the first thing you think about is Parkinson's disease, because it's very clear cut. And the cell type that you need to replace is a dopamine neuron. That's very clear cut. And there was history, and that's a huge advantage in both neurobiology and also in diabetes, that people actually tried to treat these diseases with cells that they harvested either from cadavers or fetuses. And sometimes that worked. So that's a big history lesson here. So we have that to go on. The Aspen was founded after there was a hike to the top of Mount Kilimanjaro by a nonprofit, which became a foundation, to raise $200,000. That was the goal. They raised $200,000 in 2012. Then I would hire a postdoc to work on this project. So the person I hired was Andres Bratt-Leal. So he joined my lab because of that. And so gradually my lab got more and more focused on that. We got funding from STERM. We got a little bit of funding from the NIH. we got funding from the nonprofit that originated this whole thing, the Summit for Stem Cell. They actually ended up contributing $3 million. So I was recently on a program about, or a sort of a panel about how you spin out a biotech company from an academic lab. So I don't know anyone else who's done it this way, so I can't advise it. But what happened was that around 2017 or so, we essentially set up the entire project so that we first looked to see what the FDA would want to know, and then we started doing that in our lab. And instead of doing karyotyping, of course, we did whole genome sequencing because that's what my lab did. And instead of doing like marker-based studies, we developed assays for neurons that we had developed for pluripotent stem cells, an assay called PluriTest, which allows you to predict the fate of cells. So we developed an assay that would predict that a cell at a certain stage of differentiation in a culture dish would go on to make a dopamine neuron that could connect with the brain of at least a rat. So in 2008, a lot of things were happening. Scripps was downsizing at that moment. I was actually going to end up losing my 5,000 square foot lab and have to move to a shared lab somewhere in the basement of the Beckman Institute, which is because everybody had to do that. And so it seemed to me that was a little bit of a push. And then the pull was that I got some venture capitalists interested in investing in a company. And since I had already been in biotech, and I already had a lot of connections with biotech through my collaborations with Illumina over the years, I first turned to the people who had invested in Illumina. And amazingly, they said, yeah, sounds like a great idea. And I got the seed funding from one of the first companies called Domain that had invested in Illumina. And I got $7 million altogether. But the other two sort of remarkable ones was Arch Ventures, which is a really big VC, invested in Illumina, but also a couple of other people who did. And one of them was Jonathan Silverstein at OrbiMed, who I met accidentally because he has a relative with Parkinson's disease. And he was just calling me because he liked the way I explained science. So he was calling me long before I started the company. But then when he actually heard the whole story, he immediately wanted to invest a couple $1 million in it. And the other person was Sam Altman. Sam also had a relative who had Parkinson's disease. He was intrigued by the explanations I was giving about what you could do with science. You know, it wasn't science fiction, what stem cells could do. So I went up and visited him in San Francisco. This is in 2018, so before anybody knew who he was, really. And his family invested in my company. And all of those investors have followed through in the subsequent rounds. So the amount of money that's now been raised, and of course, there's an effort being done right now to raise more because you always need more when you're in clinical trials, around $245 million.
Holy.
So $7 million for my startup. It seemed like a lot of money at the time. Cool. 70 million for the Series A, and then 145 million for the Series B. And that happened a couple of years ago. So now, in the worst of all possible times, we're trying to get another round of funding. And we will get funding. I'm absolutely certain that Aspen will get funding. The question is, how much and how long will it now take to do the next phase of this clinical trial for Parkinson's? And as Arun mentioned, the first patients that were treated, just the treatment is essentially they grow each person's iPS cells. We do a quality control on those so that we do whole genome sequencing. We make sure they pass PluriTest, and then we differentiate them into dopamine neurons. But along the way, as they're becoming dopamine neurons in a culture dish, there's a perfect place. There's a sweet spot where we know from the rat studies that they do a really great job of engrafting and also connecting with the host brain. So we narrowed down that time range. And we created a tool very much like PluriTest to characterize those cells, and that became another one of the release criteria for that stage of the manufacturing process. So everything was designed with using genomics as much as we could in order to make it a simpler and more reliable way of making sure the cells were on the right track. And of course, we also did whole genome sequencing to make sure they hadn't, like all cells do, accumulated anything important. So it's an autologous therapy. We're the only ones who've done a clinical trial like this for an autologous therapy. And even Shinya Yamanaka said that's going to cost too much. It turns out if you use these genomic tools, it doesn't cost as much. And one of the reasons it's less expensive is that you can fail fast. Let's say you have an iPS cell line, you grow it up, you do whole genome sequencing or something like that, and you haven't even started to differentiate it yet. It was grown GMP. You have lots of clones from the same person. You look at a GMP-derived clone and you say, oh my God, it's got mutations in it. We're just going to throw it away. We're not going to make a 5-liter flask of it like people have to do for an allogeneic therapy, right? So that means that there's a lot of stuff thrown away, but that means there's always a clear road. Every single patient who's been treated, there's been a clear road from their iPS cells passing all the criteria, all the way up until the time they get frozen down. And then the cells and the patient go to a surgery suite that has an MRI guided surgical apparatus. And there's a tiny little hole bored in the back of a person's head. And I do mean tiny. They don't detect it. You don't even lose any hair over it. And a catheter is inserted, guided by MRI. The cells are deposited right in the right place, which is a part of the brain called the putamen, which is the one end of the circuit that gets broken when the dopamine neurons die. So we're putting dopamine neurons straight in there. They're young when they start. And I predicted, because I'm just a scientist, so who cares what I think, right? That they would start feeling better after 6 months. Unlike other people who'd used allogeneic and had to immunosuppress people, these people were not immunosuppressed. So I thought, well, they're healthier. They're their own cells during development. Cells recognize cells. So I wouldn't be surprised if they started being better at 6 months. And bingo, they're better at 6 months. So that report that Arun just heard was on 6-month data. They'd only had those cells for 6 months in their brains. And almost all of them do well. There are lots of things I can't control, the scientists can't control when the cells leave the Aspen. And all the manufacturing is done inside Aspen, by the way. There's no outsourcing of anything if we can avoid it. We're even going to start, and when I say we, I mean the people who are doing it, are going to start insourcing even gene expression profiling, because outsourcing, there's always some risk. They'll get them mixed up. It'll take them 3 months to get your data back. We don't want to have to deal with any of that. So there's a GMP manufacturing site at Aspen. All the cells are manufactured there. There are lots of automation things being tested now to automate different processes of it so that you can have fewer people that you have to pay to watch over the cells all the time. And so those cells go to the surgeon. They get injected. They get pulled out. It's a little bit they don't get pulled out. The catheter gets pulled out, and then that person can actually go home the same day. Wow. They've been keeping them for overnight for observation just because that's what the protocol decided to do. And we weren't supposed to be reporting outcome measures until a year, as far as the FDA is concerned. So reporting at 6 months is just like, we could not do that.
Yeah, particularly when the news is good, right? And this is you, Jeanne. In the earlier question about the genomic integrity, you tell the people the hard truths they want to hear. And here you do the things that people say they don't want to do or you can't do. And you just do. You do the lift. It's a heavy lift, and you do it, and you solve the problem that's in front of you. But you got the big picture in mind. I think that's why you've done so many different things, because there's so much of interest, right? And there isn't an obstacle out there that scares away Dr. Loring. So probably the secret to your success there that you've shared. I want to drill down a little bit. This is a bit of an aside, but I can't not mention it because we live in the world, right? And this is a bit of a segue from both. Your answer about I want to highlight, by the way, the courage it takes now that a lot of people have moved on from this idea of autologous after the early trials in the
And Jeanne's lab, yeah,
labs, and like they forgot that that's the ultimate that's like the holy grail, right? And that was the whole idea. And the cost, it's not just practical. So I have to say, just my cap to you for saying, okay, we're going to do it anyway. And they say, yeah, you're wrong. You can do it. It is practical. So nice work. Not surprised, but impressed as usual. But to that point about autologous therapy and iPS cells, which was a big deal, it still is. And how that intersects. Now, this may seem a bit far field, but with DEI, right? We were at ISSCR, of course, Arun just mentioned, and across the board, big push to expand the DEI initiatives. And it was almost like comical juxtaposed with all the slashing of funding by this Trump administration. I mean, it would be comical if it weren't so tragic and damaging to like our fundamental fabric. But you kind of sometimes have to laugh if you won't cry. I mean, I was listening on the radio how they canceled a bunch of grants because they involved transgenic mice. Oh, God forbid, transgenic mice, not even getting it. And I mean, my own experience, I don't want to drill too deep because God knows if someone's listening, but we were talking about on a webpage about equitable outcomes for all patients, had nothing to do with anything but equity. And so our website was flagged and funding was held up. I mean, real consequences. So ridiculous and tragic, but sometimes you almost have to laugh. But I'm talking really to you about this forum piece. So getting back to the autologous stem cell thing, you've authored or part of the authorship of this forum piece in Cell Stem Cell about establishing a repository of diverse iPS reference lines, right?
It said diverse right there in the title.
We're
yeah, who knew? Timing was so perfect.
It was in the title, Jeanne. You should have known better. But
we wrote it before, but we should have been paying attention.
Yeah, I doubt that you're shying away from using the words nowadays, Jeanne. You have very few F's left to give, and that's why I love you. But the point of this story was highlighting I shouldn't say that. I mean, it's important. You got to be careful, Jeanne. You, of course, of all people, maybe I have to be careful. So I retract. But the point of the story was you were talking about real health benefits, like concrete benefits, not about like, diversity for like, whatever other reason that the administration hates. So tell us about this. And then we touched on it a little bit. You got to worry a little bit about the chill on research that any mention of DEI, as we just mentioned. How do you think the trainees have to, what's if you had to give some advice to trainees who have to be, of course, more mindful about how their work and their lives will be funded. How are they supposed to navigate this, that they can't put these words in the title? Just kind of tell us about the story and what your big picture thoughts are.
Yeah. One thing I think I've always been good at, and I didn't realize how important it would be, is saying things in different ways. I mean, making things ambivalent. And this one was out there. The first diversity cell bank, iPS cell bank I made was in 2000. We started in 2009, I believe. It was funded by the Gates Foundation that wanted diversity because our argument was drugs don't work the same for different people of different ethnicities. This is a fundamental problem. And then I was involved in the H3 Africa project, which was essentially trying to get equity in healthcare for people in Africa. And we were focused on stem cells, which was quite a reach, but we did it anyway. We still have that bank. And it is, I think it's going to have a comeback because we need, whether we call it diversity or not, we need more genetic variation. That's the way I get around that term. Mm-hmm. We need more genetic variation because drugs work differently in different people. And there's also a tendency to have different genetic backgrounds affect different degrees diseases in different people with different ethnic backgrounds. And of course, most of us are mixtures of all sorts of stuff. So That makes it a little more complicated, but I think eventually, each individual could be assessed for whether a drug will work or not.
Mm-hmm.
So what I would advise a student right now would be to go ahead and do this, but do it quietly. Do a comparison of a few cell lines. Don't make it the very big deal. Just its consistency among iPS cells, differentiating into X, Y, or Z, and then do that. Analyze it to death. That was the one thing I would say to all my students in my lab. Just analyze the shit out of it, and then you'll either find something that says they're all the same, or you'll find something that makes them different. So that's the kind of project I think would be really helpful right now. Obviously, for the Parkinson's project for Aspen, it's just people with Parkinson's disease, and since they're all individuals, they could be any racial group, they could be any ethnicity. Of course, they end up, most of them, being white. And that's because people have a higher standard of care if they're white. Mm. They're the ones who go to the neurologists who recommend them for the trials. So there are all sorts of layers that are going to be challenging. Now people are talking about using organoids made out of iPS cell-derived cells. And there will be someday, I can't say when, a market for organoids that can be used by pharma companies to screen drugs for efficacy and toxicity. They've already been using them for toxicity. And so there will be a need, and probably a very quiet need, depending on when they start. If they wait until Trump's term is over, then they can probably not be quiet about it, but they could be doing it quietly right now. Just setting up the assays that you could use to see whether they agree with the animal studies that you're doing, or disagree with them, or better predict the efficacy of a drug in all people rather than just a single ethnicity. Because some drugs have been shown to have very different effects in, like, the Chinese population. So if you do your clinical trial in China, you get this answer. But then if you come back and try to use that same drug in the US, it doesn't work on everybody. It only works on people with Chinese heritage. I mean, that's kind of a blatant kind of example, but that's what we have to deal with. There is diversity, and so we have to embrace it, even if we don't call it that.
Yeah, no, very, very well said. And, speaking of science that's on the chopping block right now, Jeanne, you're, of course, also a fellow space biologist, and it's been fun running in the same circles as you, whether it's on the stem cell side, the space side. We've run into each other quite a bit recently. And, as a space bioscience aficionado, I was super excited to see your recent publication on the effects of microgravity on human iPS-derived neural organoids on the ISS, the International Space Station, work that was, of course, led by a good friend of mine and colleague, Davide Marotta, who's with the ISS National Lab now. And so tell us more about this really cool project that's literally intersecting all of your interests. And
well, if I send rhino neurons up, then it will intersect everything. I'm working on that one. Yeah, so we started this right at the worst possible time, which was just before the pandemic started. And we really did not. We had funding from the National Stem Cell Foundation, which is very interesting and very, I can't use the word diverse. They give money to a lot of good causes. The CEO lives in Kentucky. There's a lot of space stuff going on in Kentucky, and so there was a movement to start doing things that were more related to space. So I said sure, and so we designed this organoid experiment in which we wanted to make organoids that represented parts of the brain that were affected by neurodegenerative disease. So, not a surprise, Parkinson's disease, dopamine neuron organoids. The other one we chose was multiple sclerosis, and there's a sort of more generic kind of organoid you can use because it doesn't really matter what kind of neurons they are, which is called a cortical organoid. So we had these two kinds of organoids, and we figured out after the first flight, and I think you have the same experience, Aaron, the very first flight was not a total success, but we learned a lot from it. We decided we had to simplify the system, and we ended up, for the work we ended up publishing, was we put each individual organoid into one of those vials that you use to freeze cells, a cryovial. It's just a vial that you can really screw the lid down on hard in 1 ml of medium. And we discovered that the cells will survive for months under those conditions because there are very few cells, lots of culture medium. So that's what we did for the flight that we just wrote about. We sent individual organoids up in these cryovials, and then we individually analyzed those organoids that had been on Earth and the identical set that had been on the ISS for 30 days. And the key method that actually gave us the most information was gene expression profiling, which is not a surprise. It's like all the biomarkers at once. We found that biomarkers that represented cell division were lower in space or in microgravity than on ground, and genes that are indicative of further maturity of neurons were higher in microgravity than on the ground. Those two things are consistent. When neurons develop, they stop dividing. So, and we can't tell you how much more mature they are. They were just discernibly mature. So we've had a total of 6 missions so far. And you'll be familiar with this sort of thing, Arun. We're now writing up the ones that we did 5 years ago instead of 7 years ago, because and Arun will attest to this. It is so much fun. Mm. It's really fun to take those samples to the Kennedy Space Center, to have them put on a rocket and to watch that rocket launch. Mm-hmm. I mean, there's nothing like it. And that's the reason Arun and I think are such good friends, is we have this shared experience. It is just really cool. It's really worth whatever it is to be able to do that. I think long term, I think Arun would, I don't know what Arun says when people say, well, what good is that? But I'm going to say we don't know yet, because we know a few things are really good in microgravity, work much better, but they're not biological. Most of them are not biological. Protein crystallization is much better in gravity and microgravity than it is on Earth. And the cells are clearly different. So the question is, can we learn something because the cells are different? Yes, we will. Will it be useful? We'll have to find that out. We'll definitely learn things, though. That's the great thing about adding a variable like this, is that you know that things will be different. You just don't know exactly how.
Well, yeah, that's the great thing about a good question and a well-designed experiment, is you get a great answer out of it. And you both of you guys have been putting together some novel and interesting findings from this outer world. And I got to say, I am so jealous, because it sounds like so much fun. You guys have the connection, but also that is such a visceral thrill. So I could see it. I can kind of see it vicariously through you guys.
I'm looking forward to when Arun can go up there and take the samples.
Oh, man, that's what we're all lobbying for. He's been waiting his whole life for this moment. Arun, you ready?
Fingers crossed. Let's see.
So one thing I did get to do, Arun, other than just going up there, is that in our last several flights, we used my own iPS cells to make the dopamine neurons. And
nice.
So
awesome.
My cells have been to microgravity, to space, even though I haven't been.
So you're an astronaut, Jeanne. There you go.
I'm an astronaut. I am. Yeah. I'm sort of like
You have an avatar in space.
I have an avatar astronaut. Yeah.
Although I would not rule out the white rhino. I mean, that's going to be great for a headline. Get the rhino up there, and somebody's going to make a gag out of that one. And who knows for what use? But hey, the question is worthwhile.
I agree. And, if I had full control over this, and I could just have okay, you could have 10 samples and we're not going to ask you what they are, I would do
it. The zoo in space. There's the headline.
Yeah.
But, that apropos, when we talk about your career and your life, it really is unique, really one for the books. And not coincidentally, you're having a book written about you. Just quick synthesis. I mean, what I love about it is there's nothing like the humble beginning story, right? And you were kicking around a bunch of mining towns as a kid, not some elite, East Coast or coastal education. But from that and your curiosity and brightness, you had an ascendant career, including development of cell therapy for Parkinson's disease. And this was before human embryonic stem cells were ever even derived. So you were in the cell therapy game before anyone was talking about it, before stem cells were even a thing. And as we've discussed ad nauseum here, your research interests in the intervening years and accomplishments have run the gamut. But the thing about you also that's so fascinating is you've just been in the room, right? You've been in the conversation surrounding so many of our field's pivotal developments, be they related to like the ethics of funding. You were in the middle of that heated, a little bit of a fracas there, the IP landscape. Like you took a I wouldn't say a divisive position, but you fought for what was I think you took a tough position and you won, but you really understand the whole organism, as they say, and that's not even to mention your adventures outside the lab. I love in your Wikipedia page there's this note about how you've logged 45 minutes in aggregate in the totality of solar eclipses across the globe, which, I mean, is there a cooler fact than you sent your avatar to outer space and also aggregated 45 minutes of totality? Like, wow, you're like a superhero. So as someone with such an expansive narrative arc in stem cell biology, but also in life, I want to ask you, this may be a tough one, but it's just your opinion. What's the biggest story in the relatively short history of pluripotent stem cell research? And if it's what I think you're going to say, I have a quick follow-up. What's the biggest story in stem cell?
Let's see. Good story, bad story.
Just big story for you in the moment right now as you're thinking.
Oh, right. Well, so this will depend on the time frame of your question, because the biggest
whole
thing,
history, everything.
Okay. Well, then so I made human embryonic stem cells around the same time that Jamie Thompson did. And there was a huge moment at which George Bush said, okay, the NIH can fund the ones that are already made. And I had already made some. Jamie had already made some. There had been made some at UCSF. There were some in Europe. There were some in Georgia. But it was a small number of small number of people. I mean, this is like you were there in the room at the very beginning. And that means that I got to learn from all these people. So that's probably not the answer, but that's the first one that came to mind. iPS cells, oh my God, I forgot that one. iPS cells were the biggest deal. Never mind. ES cells were just a prelude. They were just practice for iPS cells.
Well, I have to say, I expected you, sorry to interrupt, but I expect you to say iPS cells, but I love that you started with that because that was such a pivotal time. And that kind of created the whole drama that I think has been sustained, that created the sides. It was the scientists against the moderators of what should and shouldn't be done. And that is a debate that has carried through. But I did think you were going to say iPS cells because I think a lot of people would agree. And the follow-up was because that was such a game changer and kind of the counterpoint to that division, right? Because it flattened the earth for all of us. But the follow-up, I was going to say, given iPS is, most would agree, the biggest story to date, what's it going to take? What is going to be the story where it's like, okay, that's now has supplanted iPS as the biggest story? Clearly something therapeutic, but what is it?
You mean that has nothing to do with iPS cells?
No, that's something
to do with
that's stem cell related, that where it's gonna be like, okay, now the new biggest story is no longer iPS, now it's this.
Yeah, I can see that. So I'm not going to say. These are the things I'm not going to say. I'm not going to say a new form of CAR T therapy because, of course, and I'm not going to say that it'll be just some isolated cell replacement. I think the big thing that's going to happen is that people will realize that you can use pluripotent stem cells not just to make cells to replace cells that have died, but to deliver things, to kill things, as vehicles to do stuff in the body. And the reason I think that is going to be a big deal, it is actually going to happen, is CAR T therapy. Because CAR T therapy was a huge turnaround. It changed everybody's mind. I mean, it had a little bit of difficulty at the beginning, which is always a nice story. But now that it's routine, I think iPS cell-derived therapies will be too. And it will be coming back to say things like, this one is better than that one, or how much can you charge for it? Kind of not as interesting things, not as breakthrough things, which is the way CAR T therapies are right now. Somebody just asked me, it was a venture capitalist, just asked me to just look at some of the companies recently that he'd invested in. He said, well, there's just a set of ones that are doing whatever. They're all doing better ways of DNA sequencing. They're doing better ways of DNA synthesis. I met a lot of those folks at the SynBio Beta conference I went to a couple of months ago, which was like this is one of the things I love about this. I'm completely amazed that there are lots of things going on that I knew nothing about. And so just showing up in a meeting in which nobody knows anything about stem cells, but they all know about DNA and synthesis and synthetic biology, I just had the best time. And I have made a lot of friends there who are calling me up now and saying, oh, help me with my company or help me with my company. So I think to try to bring myself back to where I was, I think that there are a lot of opportunities to use stem cells in ways that we haven't used them before. And I'm not talking about cosmetics. I'm talking about fundamental things that are now not well managed. And everybody should have their iPS cells made. I mean, come on. I made mine, and so they got to go to space. Why would you not do that?
That's enough motivation for me right there. Yeah, I might have to do the same. Well, Jeanne, we got to wrap up here in a couple minutes. But before we let you go, I just wanted to ask you this question since we're so happy that you are a scientist. And I can't imagine a world where you're not a scientist. But in that world where you were not a scientist, what would you be doing with your life?
I would probably be writing novels. I don't have time for it right now, but I think I could do that.
What's great about that is that novels are the fuel for imagination. Arun and I are always geeking out about sci-fi. But you're like this, again, a superhero who draws the narrative out of the biology, right? Like you see the bio and then you want to know the story and then you tell the story. And you might argue that is better than a novel. Fiction, why take fiction when you can have truth, right?
Mm-hmm. Yeah, enough truth.
And finally, for the trainees out there, you gave a bit of advice on how to navigate this DEI quagmire. But what would be the greatest piece of advice you've ever been given, either professional or not, that you would share?
Yeah, so there's lots of them. But I think now that a lot of our youngsters, our graduate students are going into biotechnology companies, I would say that you need to retain your academic spirit in a biotech company, no matter how hard it is. That the quality of the research has to be better in biotech than it was in the academic lab. And you have to be more careful. And you can't rely on that biotech company, I can speak from my own experience, to support you the rest of your life. It's going to be your accomplishments at that company that will lead to the next job that you have on the next one, whether it's an academic job or another job in a biotech company, because I've been in both. They're great. I like them both. But the one thing you have to do is maintain your own self, your own reputation.
That's a great one. Because coming from you in particular, who's navigated both worlds, that's not obvious. But now that you mention it, it's so important, right? Because I think that is a perception. And as more and more people move out of academia and into industry and then the stem cell field, because the opportunities are there, the perception of like you're switching one thing for another needs to change, because it's still you, right? It's still you. Wherever you go, you gotta drive the work and the innovation. So that's a great one and a great way to end. So thanks for sharing it all, Jeanne. Like I said, we can't get to all of it with you, and we could go on for hours, but we're going to have to just run the tape next time we run into you at a meeting, and then make a little podcast series, perhaps on the ground with Jeanne Loring, on the ground and in space, perhaps with Jeanne Loring.
Yeah, maybe we can bounce it off, through the astronauts and on the space station as it goes over.
Well, Arun's Rolodex has all the names in there, so we'll make it happen.
That would be fun. I will be, I'm going to go to Montreal next year. There's no way you can keep me away from there.
Looking forward to seeing them, and maybe even before then. So yeah, on behalf of our listeners and Arun, thank you again, Jeanne, and we'll talk to you soon.
Yeah, thank you both. It's good to talk to you. Bye.
All right, that brings us to the end of this tricentennial episode. Thank you guys so much for joining us. If you're joining us for the first time or after 300 episodes, don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCELLpodcast.com, or via email at info@stemcellpodcast.com with feedback or to suggest guests. Thanks again so much, you guys, for listening. We'll be back again in a couple weeks with a new episode, number 301, starting off a new cycle.